Recombinant DNA, learn to draw the diagram showing the formation of a recombinant DNA Draw a horizontal double helical structure at the top left end of the page. This is the coiled part of DNA. Now on the right side of the DNA, draw a rectangular box and write base sequence GAATTC on the upper strand and the complementary sequence CTTAAG on the lower strand. Label the complete structure as vector DNA. Similar to the first structure, draw another horizontal double helical structure at the top right end of the page. Draw a rectangular box and write the base sequence GAATTC on the upper strand and CTTAAG on the lower strand on the left side of the DNA. Label it as foreign DNA. Draw two arrows between the bases G and A, one on the upper strand and another on the lower strand of the uncoiled part of the vector DNA. This represents the ECO-R1 recognition site. Similarly, draw two arrows between the bases G and A, one on the upper strand and another on the lower strand of the uncoiled part of the foreign DNA. Draw an arrow pointing downwards. Again draw the vector DNA. However, in the uncoiled part, do not draw the portions to the right of the arrows. Label this remaining part of the uncoiled DNA as sticky end. Similarly, draw the foreign DNA, leaving out the portion to the left of the arrows and label the remaining part as sticky end. Draw an arrow showing the joining of the sticky end. Draw an arrow pointing downwards. Now draw the vector and foreign DNA in such a way that the sticky ends of the two different DNA molecules fit on each other. Write recombinant DNA below this. Here is the label diagram of a recombinant DNA